Hello, everyone, and welcome. Today we're doing something new. We have a new series and a new format. Ever since the COVID-19 pandemic started, I have not had access to a classroom. Therefore, I think I'll be switching over to virtual for the time being, as we're still in this pandemic. And if I like it, we just might keep it this way. But today we're doing something new. I hope all of you are staying safe and healthy to the best of your abilities. And it's only fitting, I suppose, that we're going to talk about immunology in this time period. This is a new series, an immunology series. And immune system is a very interesting system. It's a big conglomerate of a lot of other disciplines in the body that you have going on: hematology, molecular biology, physiology, some anatomy, some histology. It's everywhere. So it can be a a rather interesting subject. Can be challenging, but just give yourself ample time to digest it. I hope all of you are able to reach your goals in that regard, in every regard, really, assuming you have good goals, of course. Uh, but that's aside the point. So, every story has a beginning, and that's really what our lecture is today. We're doing an introduction to immunology. And you can't really talk about immunology without talking about the antibody or the antibody. The antibody really is just a serum protein. The serum just being, that really it's mixed in with your blood, just that protein component of your blood. And there's different types, but just in the general sense, what you expect to have from an antibody is going to be a variable chain and variable region and a constant region, and then interspersed and has sub names, if you will, called a variable region and a constant region on an antigen binding domain and a constant domain. I think that's a mouthful. I don't want to confuse anyone. But here I'm just drawing what we have here, what's called the heavy chain. And so just remember, it has different subunits as a lot of proteins do. And if you don't know that, now you know. And if you don't know that, there's no shame in that, but you learn. Um, so this is the heavy chain, and then um, um, we'll do this light chain here. Okay. And so I'm just drawing these. So it's basically different proteins kind of complex together into a bigger protein. That's essentially what a subunit is. And then we'll just draw in here, these are disulfide bonds. It's a bit slow. Sorry, I'm still getting used to this format. Okay, so just to go over everything in this sense. So this is the antibody. It's a curious looking thing. Looks like a Y, but then the Y has like another two extra prongs like floating on top of it, and that's the light chain essentially. Okay. So there is a variable and constant region. So, and this is the variable heavy chain because we said the blue is the heavy. So this is variable heavy, and this is variable light. Okay, and that's just the same on the other side. And then you have constant light. So this is the constant part of the light chain. So all this red thing is the light chain, and the heavy chain is down here. And then so you just have the constant heavy, and so all these are constant heavy as well. All these are constant heavy. Okay. And then there's two domains on here you should know about. Or I think you should know about. Well, I already used green, so let's pick a different. Let's pick pink. You have this part, right? This is called the antigen binding domain. Some people call it FAB. This is where antigens bind. And what is an antigen? In the general sense, an antigen is just a protein structure. So truly, a lot of things in a lot of things in this world can be considered antigens as long as it has some kind of protein. Now, there's such thing as lipid antigens, but that's really not the mainstay type discussion that we're having. But antigens are recognized by these antigen binding regions of the antibody, right? And then there's this region down here. This is called the fragment crystallizable region. But because C is also for constant, some people also say this is like the constant region. There's truth because the heavy chain constant is down here, but truly just it's crystallizable fragment, just some biomedical jargon, but that's what it means. But that's what it is. So sorry if I bogged you down a bit. This is just a general structure of an antibody. You have this variable region that binds antibody. So as you can see, because it's like a Y, actually one antibody of this type can bind two antigens, right? And so this actually rudimentary. There's actually an IgG antibody because it's a monomer, but we'll talk about this later. So yeah, so just think in the big sense, antibody binds antigen. So it's just one protein that binds another protein, and these antibodies are made by our own bodies, right? And then they bind antigen, and then they do different things to the antigen, and we'll talk about them. But so there are different classes of the antibody, right? The antibody could be IgG. This is the most common in the blood. It's about 75%. And this is a monomer, so that means it's just one of these Y thingies, right? And then you have IgM, which is actually a pentamer. So the pentamer is actually, it looks like this, but 5 times. And so you might think, well, what does that even look like in that regard? Try to sketch it out here. It actually just, what is this? It just looks like looks like a snowflake. So you have like one Y, you have another Y, then you have another Y. Okay. Then you have yet another one. So yeah, it's going to be 5 total, and therefore sometimes they can call IgM pentameric. And so you can actually see that this means it has 10 binding sites. So IgG has 2 binding sites. IgM, because it's actually 5 times 2, it's 10 binding sites. And you have something called IgA, IgD, and IgE. These have discussions on them. We'll have discussions on them later. But just I really stress the antibody because it's really the mainstay of the immune system, or one aspect of the immune system. So how do you know if your antibody is going to be IgG, M, A, D, or E? It's dependent on the genetic coding of the Fc region. So the Fc region is going to determine whether or not it's going to be any of those different classes, right? And then remember, the antibody is it's structurally the exact same thing as a B cell receptor. B cell is one of the type of white blood cells that you have. And they're actually the ones that produce antibodies. So it's interesting that when they produce antibodies, literally what the antibody is just their receptor untethered in a sense. So they have some receptors on their surface, and then they're also going to give off some of those receptors to float freely, and those free floating receptors are what the antibody is. And so one B cell can produce one type of antibody because one B cell recognizes exactly one type of antigen. It is very precise. And so we'll look at some numbers later that kind of contextualizes the magnitude of what we're talking about. Oh, so this doesn't clear all drawings. Very good. So if we said that one antibody only recognizes one specific antigen, how do we make sense of this? So we have 100 million types of unique antibodies in our serum. So that's 10,000 heavy chains times 10,000 light chains, making 100 million unique antibodies. You know, those are combinations because it's a light chain and the heavy chain kind of put together by the disulfide bonds. But we only have 25,000 genes per cell, right? So in our cells, you know, inside the nucleus, you just have a lot coiled up DNA, and the codes 25,000 genes. But we have 100 million distinct antibodies, so it's clearly not going to be one gene per antibody. You know, with all those antibodies, we'd run out of genes, and we do. So there was a mystery until 1977 when you have Susumu Tonegawa. He's a scientist, PhD scientist, yes. And he found that DNA sequencing in an immature B cell light chain was different from that of a mature B cell. So this means that, deduce that an immature B cell's light chain is made by the genetic code that we have. And then when the maturity of the B cell happens, the genetic code is different on that light chain. And so this means that the maturation process itself yields genetic diversity. So it's actually going to mean that we don't need 100 million genes. Rather, we just need a base set of genes. A process can occur that can scramble up those genes and make more genes from those genes, right? And so that's really why this was quite revolutionary. You know, Dr. Tonegawa, he's still alive as of I'm making this video in 2021. He's 81 now, so props to him. And he made this excellent discovery. And so the mechanism for this diversity is I kind of touched on it. There is a germline DNA. So just our DNA is the germline, the baseline DNA, right? And it has different segments, a V, D, J, and C segment. And then a process called recombination or somatic recombination occurs. It's going to splice up the V, D, J, and C segments. And paste them together, or paste certain fragments together and make paper mache of VD, J, and C segments. This paper mache is directed by RAG1 and 2 enzymes. But when you mix and match this germline into different sequences and mixes and matches and whatnot, you're actually making different receptors. And so when you do this 100 million times, you're going to code for some receptors that recognize antigens that are important for the immune system. That being said, because it is a mix and match of 100 million random times, theoretically, we do have antibodies for antigens that don't even exist, right? So that's just the nature of this, but some will just chance alone, you will have a practical receptor for it. So remember, the antibody will bind its cognate antigen, which is a precise one-to-one match. One antibody can only match its antigen, so it's a very precise binding in that regard. So 100 million is quite useful. Very good. So now we move on. So here is the going over what we said before, just in more detail. In our germline DNA quote-unquote toolkit, we have 40V, 25D, 6J, and 10C segments. Recombination will occur via RAG1 and 2. It'll mix and match some of these segments, put together different, V, D, J, and C segments, and then it will make eventually around over 100 million type of codes for a receptor. Right, and so again, that process is called somatic recombination, and then junctional diversity is just the name of a process, but essentially when the receptor undergoes its junctional diversity, it's like a tidying up the receptor. Some DNA bases might be added or removed just to make sure that it is a functional protein, just tidying it up. Okay, so it's trimmed down and tidied up now, and then now you have you do this for light chain, you do this for heavy chain, and now you have a B cell receptor or an antibody if it's not stuck to the membrane of the B cell. So let's do some more food for thought. If there's 100 million types of B cells, there are 3 billion B cells, and there are 3 billion B cells inside of you at this very moment. You divide 3 billion by 100 million, and that's only 30. So for, let's just say, antigen A, just an arbitrary name, you only have 30 B cells that can handle it, assuming, antigen A is like harmful and whatnot. But you'd probably need more than 30 cells, and mind you, these are super microscopic little cells to work on this antigen, right? So what happens? Something called clonal selection happens. And so once an antibody is made, it is tacked onto a B cell surface, and the B cell wanders the body, right? And then the B cell is going to wander around with its B cell receptor in the lymphatics usually. And if it comes across that antigen, most won't, because again, most antigens that we have receptors for probably don't exist. But if it does find it, the B cell will start proliferating. So it'll make lots of clones of itself. And suddenly you go from 30 to 20,000 or more, you know what I'm saying? So if a B cell finds its antigen, corresponding antigen, it will proliferate, so make more of itself. So you have more than just 30 cells handling a possible infection, right? So we just talked all this antibody. What does antibody do, right? Why open our lecture on immunology with antibodies? This is an antibody class, it's an immunology class. So why? Well, because it has some important function, and also we revisit antibody throughout the series because antibody is recurring and super important. So the three main ones we talk about is opsonization. It's a funny word. Maybe you do not hear it before because I don't think it's used outside the context of immunology too much. But basically, if the free-floating antibody's antigen binding region latches onto a cognate, to rephrase, the antigen binding region will bind the antigen, right? But then if you imagine it's like a Y, right? That leaves the FC region kind of like it's hanging out there. So let's just say this is antigen, and then this is antibody. So this is FC region is binding right here, right? So this is FC right here, right? Sorry, FNAB, right? And then that just leaves the FC region out here, right? So what happens to the FC region? Does it just, stay wandering and whatnot? Well, not wandering. Rather, FC region looks like an L. The FC region has corresponding receptors on some white blood cells. I'm not very good at this. Has some white blood cells. So other white blood cells, like namely the macrophage, which is a big eater, hence the name. It's another type of white blood cell that eats things, hence the name. It has FC receptors. So if a macrophage detects an FC receptor, uh, it will basically grab onto the FC receptor and then drag in the receptor into itself. It'll phagocytose it. And so because FC and FAB are connected and the antigen is stuck to the FAB, by extension, the antigen also gets dragged into the macrophage, right? So it's like if I'm going down, I'm taking you down with me, right? And so that's what opsonization is. It makes the antigen easier to be eaten via like this very sadistic, if I'm going down, I'm taking you down with me type process, in which the macrophage grabs the FC region, drags the whole antibody and the antigen, which is stuck to the antibody with it, right? That's opsonization, right? So it makes things easier to digest. It makes antigen fragments and whatnot easier to digest by macrophages, who can eat and destroy harmful things, right? And we'll talk more about those later. And then the second function is called neutralization. So if a viral protein is outside the cells and it's drifting about, as viruses tend to do, the antibody can stick to the virus, because the virus has protein components too, right? The antibody can bind to the virus and basically render it useless, right? So it just like clogs up all its binding sites or whatever, changes up its configuration so that the virus is rendered, neutral. It doesn't do anything, right? So it just clogs up all viral function in a sense, or most viral function, depending on the virus, right? And the third thing is it's a scaffold for complement system activation. Complement system is another protein system we will be discussing in a later lecture. But there's something called and again, it's going to sound weird now, but we can talk about it later. It's called the classical pathway. Complement are proteins that destroy pathogens, right? And so one way that complement knows like what to attack, like what is a pathogen, is that antibodies basically serve as a scaffold for those complement proteins to come in and dock onto the pathogen surface to do their thing and destroy it. But we have a whole like lecture on complement. So let's clear here. Let's switch to mouse. I said let's clear here. Okay, let's switch to mouse. Okay. So what's the limitation to B cells and their antibodies? They're great. They're really good. But an antibody, like we said, just like in your serum and your plasma. So if an antigen on an invader leaves the plasma and goes inside a cell, like viruses, like they just leave the serum or the plasma or the extracellular space or whatever, and goes inside a cell, how does the antibody find them to do neutralization, opsonization, scaffold complement? Well, it doesn't, because complement doesn't go inside cells, right? It doesn't follow virus inside the cells. So therefore, the body needs another mechanism in order to deal with antigens and viruses and whatnot, viral antigens or bacterial antigens that are hiding inside cells, that not accessible to antibodies. And that's why we have the T cells, right? So that's I put mnemonics throughout here. Some people might find them a little bit, I don't know, unnecessary, but I have the mind of a 5-year-old. I need mnemonics for everything. Think of T cells as very gossipy cells that they need to know deep into the lives of others. That's how T cells look inside, right? So how does the T cell look for antigens inside other cells? Does it like rip the cell open and look inside? Well, no, because you'd kill the cell if you ripped it open. So there's something called MHC presentation. And MHC presentation is how T cells look inside other cells, right? And there are 3 types of T cells. You can divide them further. You have cytotoxic T cells. These are called CD8s. You have helper T cells called CD4s. And you have regulatory T cells. Of the regulatory T cells, you have iTregs, inducible T regulatories, and natural T regulatories. But we have a whole nother type of module on T cells later, but just to prime your minds, I suppose. What happened? Here we are. So you have these things called MHC molecules, major histocompatibility complexes. These are surface proteins on our body's cells, right? And they sample the cell's internal environment and project the findings, like peptides, to the outside of the cell for viewing by T cells. So all nucleated cells have MHC class 1, and then antigen presenting cells. So these are cells that are nucleated, so they do have MHC class 1, but in addition to their MHC class 1, they have MHC class 2. MHC class 2 is like a specialized MHC specifically for cells whose job it is to sample the world around them, break it down, and then share antigens. In this way, they can amplify the immune response. So it's not only B cells that take care of extracellular antigens through their antibodies, which are extracellular, but also the professional antigen presenting cells can take up antigens, break them down, and project them to also get T cells involved in extracellular, but also intracellular through MHC class 1, which projects the insides of a cell, as can MHC class 2 in some situations. I hope I didn't confuse you. Big thing to just keep in mind, MHC molecules are how T cells look inside cells. All nucleated body cells, so literally anything, cardiomyocytes, cardiac myocytes, pneumocytes, I don't know, neuron, anything that has a nucleus has these MHC. And then MHC class 2 is just for these specific antigen presenting cells, which we'll talk about who they are later. Okay, and so you can imagine a lot of autoimmune diseases occur because there's a defect in MHC molecules. And so actually, hence the name histocompatibility. We talk about graft rejections and like organ transplants and whatnot. MHCs are a very important topic. So what's the structural difference in MHC molecules? Try to draw it out for you, though I don't think the drawing is particularly useful for this. But if it is, then we'll do it. MHC class 1 looks like this. So just imagine this is a cell membrane. It's a lipid bilayer, but I'm just drawing one for now. And so then there's different domains to this molecule, right? So it's transmembrane, so it has like this part in the cytosol, cytoplasm, whatever you want to call it, and then it has like these loops. So it has second loop, and then has the third loop. So this is alpha. It's actually 3 alpha chains: alpha 1, 2, and 3, and then alpha 1 is tethered to something called beta 2 microglobulin, which is this. Okay, that's what it looks like. And this is different from a MHC class 2, which if I draw it out, this is just a membrane. It looks a little bit different because MHC class 2 has an alpha chain and the beta chain. So alpha 1 up top, alpha 2 is the transmembrane part. It also has beta 1 and beta 2, and then like that. So there's no beta 2 microglobulin. So you can almost think about an MHC class 1 because it doesn't have a beta chain. It's just 3 alphas. It compensates by having a beta 2 micro, like a small beta to like compensate. If that maybe helps. I don't know why test takers seem to like talking about beta 2 microglobulin. There is some clinical correlations, and we talk about them a little bit later. I believe it deals with dialysis patients, but it's not for today. But yeah, so this is what MHC molecules look like. And so what they do is that they project antigen, right? And so again, MHC class 1 has beta 2 microglobulin, and it's actually a closed groove peptide segment. And the only thing that means that it, I can do a sketch, I suppose I will. Closed groove, all that means is that it's like a cup in a sense, right? So what are we doing? So MHC class 1 is a closed groove. So it looks like this, right? It doesn't really look like this, but then the antigen will fit in this groove, right? And that's a specific size. As opposed to MHC class 2, it doesn't really have these grooves. So it's actually like not grooved, right? So therefore, the antigens that can fit inside MHC class 2 can be actually, I think, something like 3 times as long as something that fit in MHC class 1. Because yes, it will bind, but then like the ends can be tethered out because there's no groove holding anything, and it's not like a cup. You know what I'm saying? That's only the point I'm trying to make. And I have some mnemonics down here if they help you. Like I like MHC class 2. I like 2 Greek columns, alpha, beta. I don't know. You figure it out. And then because 2 is bigger than 1, it overflows. Like there's no groove, so the antigen can like just hang out. It's up to you how you want to remember that. Oh, someone didn't clear. Someone is me. Okay, and then we just wrap up the lecture now with this scary slide that's just telling you what's to come. But I assure you, it's not that scary. I hope it isn't. I really hope it's not that scary for you. It's regarding how's a T cell activated. It's a very condensed slide. I don't expect everyone to know everything from the slide, but that's all it is. A T cell is activated. It can undergo two lineages. It can become a helper T cell, which just is a cytokine factory. Cytokines are chemical that aid in the immune response, right? And they just direct other white blood cells what to do, or cause other physiological things to help the immune system. Or it can become a killer T cell, which does exactly what it says. It directly kills pathogens, right? So it's like your body's assassins, if you will. So that's a very important distinction, and we'll talk more about these later. But because a T cell, upon activation, fills very important and rather violent roles and very alarming roles, T cell activation has to have many breaks on it, because if the T cells are activated willy-nilly, you can have a very serious autoimmune disease, mind you, because T cells can perform direct cell killing. That's quite powerful. So you want checks on that very powerful type system or cell, and so there's an "are you sure" type system going on in the body, and that occurs via a mechanism in which co-stimulation is needed. So let's just take the scenario. You have a macrophage. It takes up, let's just say, bacteria. It ingests it, it destroys it in its phagolysosome, and it takes its protein fragments and projects it on MHC class 2 on its surface. Macrophage is one of the professional antigen presenting cells, therefore it has MHC class 2. T cell comes along and recognizes the cognate antigen on the MHC class 2 molecule. Okay. The macrophage knows something is wrong in the system, because there's other mechanisms to ensure that the macrophage knows that this is indeed a harmful bacteria, and to be alert. And usually it will do it through something called toll-like receptors. And I know I'm throwing a lot of words today, but after I do the second two lectures, when I talk about these things more in depth, I encourage you to revisit the slide. It'll make a lot more sense. So the macrophage already knows that whatever it just ingested is problematic. And because it knows it's problematic, it holds up a surface protein called B7. And the T cell has a receptor that recognizes B7, and that's called CD28, cluster differentiation 28. Cluster differentiation is just how a lot of surface proteins are named in immunology. You don't have to go into the biomedical science behind that. Just know that these are names, and names are names. They are made based on when discovered, so they're not really built for memorization-friendly type learning. Maybe one day the field will get standardized more and we'll have more learnable names, but in the meantime we use all these laboratory names. Anyhow, T cell binds antigen on the MHC, and then CD28 on the T cell binds B7 on the macrophage, and the macrophage won't put up B7 unless it knows something problematic is going on. When both these signals are present, then the T cell can activate. Otherwise, without this co-stimulation, T cell won't activate. Like I said, we want this break because T cells can, wreak havoc. And in the scenario I said, because it's MHC class 2 recognition, it's actually going to be a helper T cell. If it was MHC class 1 recognition, it'd become a killer T cell, but this is beyond the scope of this introductory lecture. And so part of me is scared that I overcomplicate it now. But this is, again, just a general overview. All the concepts discussed today will be revisited several times to aid in consolidation. This is just to show, what's happening in the general sense, okay? So I hope I didn't burn you out too much. That's all today. For today. Next time we're going to talk about the innate immune system. The immune system can be categorized into two subcategories: innate and adaptive. So we'll talk about innate, and in the innate we'll talk about macrophages and how they know things are truly dangerous with their toll-like receptors and whatnot. We'll talk about a lot of these things. Mind you, T cells and B cells are part of something called the adaptive immune system, but we'll talk about the difference in a future lecture. Again, this is just an overview to what's to come. Hope I didn't burn you out too much. This is my sources for today, but thank you for watching, and take care.